So dear everybody, I hope you got a cup of tea or some coffee and a cake and ready for the next talk, which is very exciting. It's one of I'm the big fan of Marla Spivak from the United States. Marla has been in Denmark. Do you remember Marla? It was in Copenhagen. I don't know, 15 years ago, or maybe it's just 10 years ago. We had
Of course, I remember. Thank you.
Because you remember the Danes. And we had a great workshop there where we really tried to focus on green breathing and hygienic behavior. And over the years, people have been asking me, Fleming, we need to get Marla back and to make a talk. And you were invited to be here in Denmark, but because of the corona, this is actually, I would say, a good solution, or the next best solution that we have. You are from the University of Minnesota in the United States, and working, as I told, about really a lot about hygienic behavior, which we are very focused on in Danish beekeeping, and very excited about to hear about what you kind of have achieved over the years, especially in respect to varroa tolerance. So Marla, I'm so excited. The stage is yours. Please start.
Thank you, Fleming. And thank you, everybody, for inviting me. I wish I was there, but here we all are at our homes behind our computer screens. So I'll give this talk, and then I look forward to the questions that you might have, and I hope that you can understand me and everything is clear. So as Fleming said, this is a talk on hygienic behavior and varroa tolerance. And I would like to begin by zooming out and giving it a bigger context. So Sylvia Kremer is, I'm a big fan of Sylvia Kremer. She's a researcher from Austria. And in 2007, she defined this term, social immunity. And basically it means how social insects maintain the health of their colony. So in her case, she refers to ants. She studies ants. Of course, I study honeybees, but it also refers to termites, shown here, or not shown, or are social wasps. And technically, the definition she gave is how these huge, densely populated societies use behaviors or organization or physiological mechanisms to defend against parasites, to maintain the health and the survival of their colony. And this is something as humans we're starting to understand very well with COVID. How do we maintain the health of our populations, especially in densely populated areas? How do we prevent virus transmission? Well, social insects colonies must think, they don't think, they don't, but they must deal with this every day of their lives. So here is Sylvia Kremer and how she defined it with her little ants. And in her case, when she studies ants, they're all in the laboratory in these little containers like this. But she said that forms of social immunity take two strategies. The first one is to take care of your nest mates, and the second strategy is to kill or eliminate them. And I know the term kill is fairly strong, so I'll use eliminate, which is a little bit nicer. So an example of a care strategy, it's like preventative medicine. So grooming, antimicrobial secretions that ants produce, and just keeping the nest clean. Those are all preventative strategies to prevent a disease or a parasite from getting into the nest. But once they're infected, then they move to the eliminate strategy, and they sacrifice individuals, infected individuals. So I'll give the example. I know this is about ants, but I hope that you as beekeepers find this somewhat interesting. This little ant, Lasius neglectus, when they get a fungal pathogen, metarhizium, because ants live in the soil, they can get fungal pathogens, and it might be exposed on the surface of the pupa of the ant, on the cocoon of the ant. And if the adult ants find this metarhizium spores on the pupa, then they start grooming the spores off of the pupa. And then they secrete a little bit of venom and put it on top of the pupa. And the venom is antimicrobial, and that disinfects the rest of the fungus. So that's their care strategy. But if that pupa becomes infected, if the metarhizium goes into the pupa and through the cocoon and into the pupa, then they eliminate that pupa from the nest. They destroy it completely. They remove the pupal cocoon, as you can see here, and then they dismember the pupa. They tear it into bits, and they put it in a part of their nest where they don't touch it anymore. So interestingly, they found, one of the students found that they are able, the metarhizium, the fungal spores grow mycelia. And when the ants are removing and dismembering and eliminating the infected pupa, they're doing it while the mycelia are growing, and they're not infectious at that stage. So it's possible for the ants to touch and handle and bite and dismember the pupa without spreading the infection, because it's not infectious at that stage. And they also then inject venom into their own pupa and before they eliminate it to sterilize it. So I find this very interesting, and I thought, okay, what are the care and elimination strategies in honeybees? What is their preventative care? And then what's their elimination strategy? Well, one of the preventative cares is propolis collection. These are antimicrobial resins, and I'll talk about these briefly later in this talk. But their elimination strategy is hygienic behavior, and that's what I'll focus on mostly. Interestingly, hygienic behavior was described in the 1930s and 1940s by some researchers in the United States. They found colonies that were resistant to American foul brood. These colonies never succumbed to American foul brood. And they wondered why, and after a lot of research, they found that some bees were able to detect and removed the disease brood, again, before the disease formed infectious spores. And then Rothenbuehler in the 60s did a lot of research on the genetics, and then later there was research on how that same behavioral defense was a mechanism of resistance to chalk brood disease, a fungal disease. Importantly, this is an elimination strategy, and as with the ants, there's a critical detection and timing component of this behavior. As explained by Woodrow and Holst in 1942, that resistance consists in the bee's ability to detect and remove the disease brood before the causative agent organism reaches the infectious spore stage in the diseased larva. So this larva may get some disease, but as these bees, the adult bees, are detecting and removing it before the infection becomes, while it's in the vegetative growth stage, not in the spore stage, and so it is not infectious, and they throw it out of the nest. So in a hygienic colony, most of the time you would not see American foul brood at this stage, because the bees would have removed it long before it reached this infectious stage. So I'll talk to you briefly about how I bred this hygienic line. This was actually years ago, and I went through these 3 steps, and I'll go through them with you one by one. First was the assay, and then propagation, and then the most important step was the challenge. So I'll explain these one by one. The assay that we did was using a freeze-killed brood assay. I understand in Europe, many researchers and beekeepers use a pin-killed assay. I think that's okay too. The purpose of the assay is to test a bunch of colonies and to narrow down the group of colonies to find those that are likely to have the genes for hygienic behavior. This is not the final test. It's a beginning assay. The rate of removal of dead brood, whether it's freeze-killed brood or pin-killed brood, how quickly they can remove it is correlated, not perfectly, but it's somewhat correlated with the rate of removal of diseased and mite-infested brood. So the way we did it was to take these plastic PVC pipes, 3 inches, I don't know how many in centimeters that is right now, but it covers 160 pupa, sealed brood cells. And then we would pour 400 milliliters of liquid nitrogen down the tube and freeze the brood hard. We'd then put that comb back into the colony and wait 24 hours and come back and check the removal rate and then repeat the test. So you may have seen these photos. These are fairly old now, but here is where the tube was. And you can see that within 24 hours, this colony uncapped and removed over 95% of the freeze-killed brood in 24 hours. For me, that 24 hours is critical. Originally, we were using 48 hours, but later on, I realized that's not quick enough. It needs to be 24 hours to find these colonies that remove almost all of that dead brood in 24 hours. If we find a colony like this, we always repeat the test. If on the second test, they perform like this, that's about, maybe over 50% uncapped and removed, they're not included in the program. We get rid of them, or we cull them, we just don't use them. So this colony, you can see they're uncapping and removing the freeze-killed brood after 24 hours, but they're very slow at it. So it's important to repeat the test. It can be repeated the next day, it can be repeated a week later or a month later, but every time, each test, the colony must perform to the same standard. If they have 2 different results, I never included them in my breeding program. We can test on a very large scale. We have many of these large tanks, Dewar tanks that hold liquid nitrogen with a valve. And we can go to a commercial apiary with lots of colonies and remove individual frames from those colonies, mark the number of the colony or some identifier on there, freeze many at once, and then put them back in the colony. You can see where the tube was. We put a thumbtack right there on the frame so we know which frame and which side it was on. Here's another frame going to go back into this colony. And then we come back in 24 hours and check, and it goes fairly quickly. Okay, once we found colonies that were highly hygienic, then I would raise daughter queens from them. And then I instrumentally inseminated those queens with 8 to 10, sometimes microliters of semen from drones from other very good hygienic colonies. So colonies that were very fast at it. And why instrumental insemination? It's because, as you know, the queen mates with 10 to 20 drones. She's highly promiscuous, and a daughter queen from a hygienic colony needs to mate with drones from other hygienic colonies for her colony to display good hygienic behavior. So for example, if a queen mates with 20 drones, at least 10 of those drones need to come from highly hygienic colonies for the daughter colony to show hygienic behavior. If you have a very good hygienic queen and you raise daughters from her and she open mates naturally with unselected drones, that daughter queen's colony will not necessarily be hygienic. You can lose the trait. Okay, so that's the assay and the propagation. And I want to say that before we even did the assay, I should say, let me say it better. We only performed the assay on colonies that we really liked, that we had followed for over a year, that we know that were gentle, that wintered well in our area because we have a climate, I think it might even be colder than your climate, not sure, but we have a very cold climate here. So they have to survive our winter well, and they have to build up well in the spring without feeding. So I'm looking for colonies that are very hardy in a cold climate. And then if they produced honey the previous year and they were low mite levels, and well, I'll get to that part later, and winter well, build up well in the spring, and they're nice colonies, and then we do the assay. Okay, all right, moving to step 3, which is the most important part of selecting for this behavior, is the challenge. So at first, when we had colonies, I was selecting for hygienic behavior. The first test we did was give them a challenge with American foul brood. And of course, after this experiment, we had to burn all the colonies. What we did was collect combs from colonies that we found in our state from other beekeepers that had some American foul brood, and we had them frozen in our freezer in the laboratory. And we would cut out comb sections with scale, American foul brood scale, which is highly infectious to other bees. We'd cut out the scale and we would introduce it into our hygienic colonies, and then another set of colonies that I was also breeding that were not hygienic. They were non-hygienic colonies. So at that time, I had 2 sets of colonies. And we let the queens lay eggs in these horrible comb sections and then rear the brood. And then we would quantify the amount of American foul brood those colonies developed over time. And so here's this photo of the apiary, and you can see that, well, you can guess these colonies were hygienic and these were not hygienic because they were dying from American foul brood. But here's the data, and these are older data, but this is the time on this axis, the time when we gave those comb sections with American foul brood scale and the number of weeks of the experiment. And this vertical axis shows the number of American foul colonies, the number of colonies that had signs of American foul brood. And I'll start with the red bars, the non-hygienic colonies. You can see that 1 week after giving them the comb section, 6 out of 18 colonies had signs of American foul brood. And by week 3, 17 of 18 colonies. I don't know why the one never developed signs of American foul brood. And by week 5, one of the colonies went queenless, and so it didn't have brood. And so that's another way to not have foul brood, but it's really not a solution. It just didn't have a queen or brood. The hygienic colonies, and this is important to realize, by week 2, 7 of the 18 colonies had some American foul brood. And then over time, they were uncapping and removing the larvae when they were not infectious. And so most of the colonies eliminated the signs of American foul brood. And at the end of the experiment, there were 2 of the 18 colonies that still had American foul brood. This graph does not show you the severity, the intensity of the infection, but that's published in this paper. And the severity was very high in the non-hygienic colonies and very low in the hygienic colonies. So they had the disease at a low level, and most of them cured themselves, except for 2. This is the same with chalk brood disease. If you challenge colonies that you've chosen based on the freeze-killed brood assay, if you challenge them with American foul brood or chalk brood disease, you will see some colonies that show signs of the disease, and over time they'll get rid of it, but not all of them. So this tells me that using this selection tool, this assay of freeze-killed brood, and I've not used the pin-killed brood, but I imagine it's the same. Just using that assay does not guarantee complete resistance to the disease. And I promise you I'm getting to varroa mites very soon. I just want to make the point that all colonies, whether they're hygienic or non-hygienic, all bees, all honeybees, can detect and remove diseased brood, but when often they do it when the pathogen is already infectious and when the stimulus level is very high. So here's a cell with chalk brood, and maybe in your colonies you've seen that they do uncap these cells, but if they remove this chalk brood mummy now, it would be highly infectious, and they might transmit the disease through the colony. Same with American foul brood. You can see the bees that have opened up these cells. If they touch the brood inside, it's highly infectious. So this trait is something that all bees can do, but some bees are very quick at it and some bees are not so quick. Okay, now getting to what you asked me to speak on. Is hygienic behavior a defense against Varroa? And this is something I asked when I first moved to Minnesota in the 1990s. I had these colonies, the Minnesota hygienic line, that I had selected using the freeze-killed brood assay, and then I wanted to know if they were resistant possibly to, or tolerant, or what they did if I challenged them with Varroa. So here's the challenge, and this I learned from Otto Becking in Germany in the 90s. Thank you, Otto. He showed me how to very gently uncap or open up a cell capping. You collect mites from different colonies. You introduce a mite into a cell that was recently sealed within 4 to 6 hours. So it's a larva that's just capped with wax. And then you mark the location of that pupa or prepupa cell on a plastic sheet. And then you put the frame into the colony, and you can come back at various intervals to figure out if the bees have detected and removed that cell containing the Varroa, if they remove the pupa from the cell. These are older data, but they're still good data. And this time the hygienic colonies in blue, you can see they removed 60 to 70% of the pupa that we infested with mites. I don't know what happened in 1995. This happens in research. You know how it is with honeybee colonies. They never do exactly the same thing every year. So that's why I repeated it in other years. The non-hygienic colonies, in contrast, removed very, very few of the mite-infested pupae. And these are the controls, meaning that we simply lifted up the cell capping, put the paintbrush in there, and then remove the paintbrush and then tamp the cell capping back down. It was a sham operation. So this showed me that just using the assay for the freeze-killed brood assay for hygienic behavior meant that these colonies were also able, the very hygienic colonies were also able to detect and remove mite-infested brood. And that yes, hygienic behavior is one defense against Varroa. And now we know that there's the similar timing component of this behavior where the bees are detecting that the cell contains Varroa mites after the foundress mite, the mother mite, starts ovipositing, laying eggs. So here, of course, is the foundress. Here's a male mite and some pseudonymphs. And this one has undergone her last molt, and she's going to be an adult soon enough to mate with this male. The bees are detecting that there's mites in the cell after that mite starts laying eggs, which ensures that any of the progeny, the mite babies, pseudonymphs, prononymphs, are killed during the removal process. The bees remove the entire pupa, sometimes slowly, sometimes they leave a portion of the pupa in the cell, but all of the progeny mites, the baby mites, die during the removal process. The mother mite, the foundress, like shown here, might escape, but eventually she does run out of sperm when she goes to go into another cell to reproduce. And so the removal of the mite-infested brood reduces the reproductive capacity of the mites. We tested our hygienic queens in many apiaries of large-scale commercial beekeepers here in the United States, and we found that they always produced as much, if not more, honey compared to the beekeepers' unselected colonies, and they had significantly lower disease, chalk brood mostly, and mite loads. And these are all older data that are published. Okay. I stopped breeding and maintaining the Minnesota hygienic line in 2009, so it's been quite a long time now. Part of the reason that I stopped breeding the line was because they became very popular with beekeepers in the United States, and they were asking me for queens, and I'm a researcher, not a commercial beekeeper. But mostly, I was afraid that if many beekeepers got the queens that I was breeding and started raising queens from them, we would lose genetic diversity in the United States. And I really think and know that genetic diversity using many kinds of stocks of bees is very important to honeybee health. So what I did instead was go to the commercial queen producers operations myself and showed them how to do the hygienic test. And now many queen producers throughout the United States, particularly in Northern California, use the hygienic test, the freeze-killed brood assay, and they're propagating hygienic stock from their own genetically diverse stocks of bees. Okay. During this whole time, another line of bees was developed, and oh my goodness, this has a typo in it. I think I did the same problem in my recording. It's called the Varroa Sensitive Hygiene, and this should say VSH and not VHS. I'm sorry if I used the wrong version of this talk. Anyway, I want to give you a little bit of history of how that came to be. This was a line of bees that was bred by John Harbaugh, and he was at the USDA laboratory in Baton Rouge, Louisiana, in the southern United States. And in the 90s, he was also looking for colonies that were resistant to Varroa mites. And what he did was measure the mite population of in a colony at the beginning of the season and then at the end of the summer. And he was looking for colonies that had low mite population growth over the season compared to other colonies. And the colonies that had the lowest mite population growth, he raised daughter queens from them, inseminated them, and he called the line suppression of mite reproduction, or SMR. He had no idea of the mechanism why there was low mite growth, but he just kept seeing it, and he found that he could select for it, meaning it was heritable. And so he continued over several generations, several years, this SMR line. And then he gave me some queens into Minnesota, and I tested them also using a freeze-killed brood test. And also we did the challenge by introducing mites into cells of these SMR colonies. And we found that they were much better at removing mite-infested brood than my hygienic line, those in blue. So here's the SMR colonies, and this is the percent removal of mite-infested brood that we introduced the mites over 10 days of the pupal period. And they removed up 85–90% of the mites, where my line removed 60 to 70% of the mites. And these are the controls. So I was very impressed. I thought that you've done an amazing thing, John Harbaugh. This is great. Only I think they're very hygienic. I think the mechanism is actually just hygienic behavior. Some of the mites in the colonies had lower reproductive success, but that was the subtle effect, the secondary effect. The main effect was hygienic behavior. And so he renamed the SMR line. He gave it a new name, and he called it Varroa Sensitive Hygiene. They did their own research in Baton Rouge to confirm what we found. They used different methods, same conclusion. He said, really the mechanism for this low mite reproduction is hygienic behavior, because the bees are removing the pupae and the mother mite and the offspring, and over time, it results in low mite population growth. To explain how they are currently selecting and propagating the VSH line, this is what they're doing, and this is important to understand. They do not use a freeze-killed brood assay or a pin-killed assay to begin with. They begin directly with the challenge. So they take a comb, a frame of sealed brood containing sealed brood, and they open up 100 to 200 cells in the laboratory under a microscope, and they determine the percent infestation of mites in the brood. So they have a starting infestation, and they take this comb and they introduce it into a test colony, not the same colony. They take it and they put it into a colony that they think might have this VSH trait. And they leave it in there for 7 to 10 days. And then they come back, and again, they take that same comb back to the laboratory, and they open up another 100 to 200 cells. Very laborious. And then they compare the percent infestation from the start to the finish. And they're looking for colonies that have much lower percent infestation of the brood after the 10 days. So, and you can see in this particular colony, they've started to uncap a lot of the brood, but they haven't continued to remove all of the pupae yet. So that is how they select. It's very, very time consuming. Some people say that the VSH trait is different than general hygienic behavior using the freeze-killed or a pin-killed test, that they're actually different behavioral traits. And I use this diagram to show a lot of confusion about this. Until we really understand the genetics, which many people are looking at, I would say there's a simple test that we need to do, and for some reason I've never done, and they've never done it with the VSH line either, but we should challenge the VSH colonies with a pathogen. We should give them chalk brood or American foul brood. And if they are able to detect and remove the diseased brood and resist that, then I would say these traits are exactly the same. It's just the VSH line is very, very quick. They're better at it than the Minnesota hygienic line, possibly because they begin their selection with the challenge and not the assay. How do bees detect this disease in mite brood? We did years of research on this, and all kinds of tests, behavioral tests, poking and prodding their antennae to get recordings from the antennae, doing learning tests of their ability to distinguish between sick and healthy brood. And this is a slice of a bee brain and neurotransmitter staining. In the end, we found that they're just very good at detecting. And of course, they use their antennae as their noses. And if the bees are hygienic, they have very high sensitivity to the odors that are coming from disease and mite-infested brood. So a bee that walks along this comb, if bees could speak, this is what I think they would be saying. Okay, that smells, sorry, here's my cursor. This smells good, okay, everything okay, everything okay. Uh-oh, something's wrong here. And then that bee might poke a hole in the cell capping, and then in this ocean of bees, she gets moved away to a different task. Another bee might come along and expand that opening, and eventually they may remove the whole pupa. Sometimes, interestingly, one bee begins to poke a hole, she moves away, another bee comes along and recaps that cell opening, caps it right back over. And then another bee may open it up again, and another bee maybe more. Sometimes bees spend a lot of time capping, uncapping, recapping, before they, and sometimes they leave it entirely, and sometimes they'll expand the capping and remove it. But we know that the bees that are highly hygienic are detecting with their antennae the odors coming from the brood, and they're able to detect and discriminate the odors of diseased and healthy brood at a very, very low stimulus level. Currently, there's a lot of research going on all over the world, from Canada, from France. This is just published from France and New Zealand, from Yves Leconte's laboratory and Alison Mercer from New Zealand. And then there's research from Olav Ruppel's lab in North Carolina with Kara Wagner. They're all looking for the compounds that are emitted from pupae that are infested with varroa mites. And I will tell you that all three of these experiments have come up with different sets of chemical compounds. And each one claims to have the exact compounds that bees produce, the pupae produce, but they're all different so far. I think this lab needs to test these compounds. They need to test each other's compounds to figure it out. The goal in all of this research is to develop, if we could figure out what are the chemical odorants, compounds, that are coming from the pupae that are infested with the mites, if we could use them in our assay, in our breeding and selection assays. For example, Kara Wagner, she took her chemicals and she sprays them on top of the comb into a little tube like the plastic tubes we use, but much smaller. She sprays some of the synthetic forms of the compounds on the cell capping and puts it in the colony, and after 2 hours, a very short test, comes back, and if the bees are starting to uncap, then she figures that those colonies are hygienic, and they would have that varroa sensitive hygiene. They'd be very specific to the odors of mite infested brood. I wonder if we should be selecting for specific cues, specific odorants, or if it would be, if the way that the bees are detecting is just because they can detect that something is not normal. So let me explain this. I think, this is just a hypothesis, that as I said, as bees are walking over these cells of sealed brood, what they detect is something additional, something that's abnormal, not normal. And they have in their brain a template of what healthy is. So they can detect with their antennae healthy, 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 and then, oh, not healthy. And I don't think it matters if it's abnormal due to varroa, chalk brood, American foul brood, or dead, or virus. I think they can detect that it's abnormal, and then they start uncapping and removing. And this is the way an immune system works. It detects self from non-self, self from foreign, and nestmate recognition in social insects, where they recognize nestmates versus foreigners, non-nestmates. So these are all, this is research that's actively being conducted throughout the world. So I guess we'll all have to stay tuned and to see if we develop an assay that's more specific to varroa infested brood, or if we continue to use a general assay. So the take home parts from this part is that if you start with an assay like freeze-killed brood or pin-killed brood, and you propagate lines of bees, you cannot expect them to be resistant. You must challenge them with a pathogen or varroa mites to see their ability to uncap and remove. And then you must take it a step further and leave them untreated, those that you're propagating, to see if they're truly able to survive without treatments. So this is what we're doing right now. I've started a new breeding program. I have not been selecting bees for a long time now, over 12 years, but I've started again with a very ambitious project. I'm selecting for many traits all at once with help from these people in my laboratory. We're looking for bees that have good winter survival, and this is one colony in my backyard. to give you an idea of our winter. And as I said, I want these colonies to survive our winter very well, have good population in April. I'm also looking for colonies that collect a lot of propolis, and I'll explain this, that are also hygienic. And I'm repeating what John Harbaugh did to begin with his VSH line. I'm looking for colonies that have low Varroa population growth over the season. So what we do in colonies with marked queens, throughout the first summer, we monitor those colonies. We're looking at brood patterns, any visible signs of disease. If they have any disease, we cull them, we're not using them. We're looking at honey production, gentleness, and we're making sure all of the queens are marked. And the next year, in the spring, we monitor the mite loads on adult bees and in the brood. Twice, in early spring and in late summer. We like the colonies to start with low mite levels in the spring so that we can watch their population growth. It's a race, but in reverse, we want colonies where the mites grow slowly, not quickly. We're taking samples of 300 adult bees and washing them in alcohol to get the percent infestation on adult bees. But we're also taking these combs into the laboratory and opening up 100 to 200 cells to get the percent infestation in the brood. And it is, excuse me, I may get some coffee. It's very, very interesting when you compare the percent infestation of mites in brood and mites on adult bees. Oh my goodness. So here's some data from our colonies last August. It's just a correlation of the percent of mites on adult bees on this axis. So 5–10% on adults. And on this vertical is the percent mite infestation in the brood. And you can see in our breeding program right now, we have this interesting set of colonies right here that by the end of the season had fewer than 5% mites on adults and less than 10% mites in the brood. All of these colonies we will cull. If they even survive this winter, I will not include them in the breeding program. So I want colonies that maintain these low mite infestation levels. Sometimes you find colonies with a lot of mites on the adults, but low mites in the brood, maybe like this one. This tells me they're probably removing mite-infested brood. But colonies that have a lot of mites in the brood, when those emerge, it leads to this huge infestation. And these are the ones that I really don't like. And you can't see these all the time just from a sample of adult bees. For me, it's important to go inside and look at the brood. I wish there was an easier way to do it. I wish someone would invent some kind of a tape that you could put on the brood cells and pull it off and then wash the pupae and the mites out to get the mites in brood counts easier. Maybe you can help me devise something like that, because otherwise it's extremely tedious under the microscope. We're also sampling those colonies in spring and late summer for virus loads. This is in collaboration with Declan Schroeder. He's a virologist. He used to be at the UK and now he's at our university, University of Minnesota. So he can help me select colonies that have the lowest virus loads in spring and in fall. We are still doing our freeze-killed brood assay, looking for the most hygienic colonies. I'm still interested in that, but we're also looking at propolis collection. And to assay this, and I'll explain why we do this in a minute, we put a propolis trap, a commercial plastic trap, on top of each of our colonies at the same time every year and remove it at the same time in the early fall. We put this rim, this wooden rim with holes in it, over the trap, and then under the cover, under the cover, because these holes let air and sunlight in, and the bees don't like that, and it stimulates them to fill these traps with propolis. We remove these, we photograph these, and we score the amount of propolis in there on a scale from 1 to 5, with 5 being very high. And if we give it to 5 people that are not beekeepers, we find that the scoring and we average the scores, it becomes very accurate. I'm looking for colonies that collect a lot of propolis because I know that from our research that it's very good for their health. So in wild colonies like this tree that was taken down, and here's the entrance, when we opened it up, and this is the inside of this tree cavity with all of the combs removed, you can see this layer of propolis that lines the whole interior of the nest cavity. Tom Seeley showed this in the feral colonies he studied in New York. He called it a propolis envelope, that they're lining the interior of the nest cavity with this propolis. All of these data are published, and the bottom line of all of this so far is that bees that have a propolis envelope that helps their immune systems, and it promotes their core microbiome species. And the reason for this is the propolis is highly antimicrobial, and it's reducing the amount of pathogenic and opportunistic bacteria and other microbes within the whole nest cavity. And that means the bees' immune systems do not have to be activated as much, and that their microbiome species can become more abundant, the core, the beneficial bacterial species inside their guts and on their mouth parts outside the bees. We know that the propolis envelope helps reduce disease loads. It doesn't eliminate the disease, it helps reduce it. And so I know that combining hygienic behavior, one genetic trait, with propolis collection, which is a different genetic trait, combining those two behavioral traits would really help improve bee health. We know that bees collect more resin when they're infected with chalk brood, a fungal disease, but not a bacterial disease, American foul brood. In other words, bees are self-medicating or social medicating. They send out more resin foragers when they're sick with chalk brood. And in our area, they're collecting the majority of the resin from the trees from cottonwood leaf buds. So resin, plants produce these resins as a defense for the plant. Bees collect them. Once the resin is inside the nest, we call it propolis or propolis. Right now, we're doing more research to understand how could we stimulate colonies to bring in their own propolis envelope. And we have these boxes that were made for us that are very rough inside. The guy that made these put deep gaps and grooves and very rough hive interior to simulate the inside of a tree cavity. Okay, so very rough. And we're measuring the immune system and the disease loads and repeating the research in colonies that have this kind of a propolis envelope to understand how rough it's enough to support bees' immune systems, so that we can tell beekeepers it's important to use these kind of boxes with not just unfinished lumber, but very rough lumber that has gaps and crevices in it. Okay, so back to the breeding program. Again, in the late summer, we're checking, as I said, the mite loads again on adults, and in the brood, we're looking for viruses again. We're actively culling colonies that do not fit our criteria. We're requeening and we're treating the colonies. Right now, we're treating those colonies, but I'll show you another thing we're doing soon. We're quantifying the propolis. We are noting the colonies that require no fall feeding, because I like colonies that store a lot of honey down into the brood nest and that winter very well. So that's how we're doing the selection right now. But those are the big colonies. But additionally, we're doing one other thing. From the colonies with marked queens that we choose, that have these low mite population growth, good wintering, all those propolis, hygienic, all these characteristics we like, we make splits, divides from those colonies, and we put them in nukes, 5-frame nucleus boxes. And we graft queen cells from our best colonies, and we put those queen cells into these nukes, and we let them mate naturally. We have an area that's very isolated from other beekeepers, fortunately, at our university. It's south of the city, and it's thousands of acres, and no other beekeepers are around. So we can let our queens mate naturally in this isolated area. By midsummer, we put another box on top of these nukes and make sure it becomes full of honey. If we have to feed syrup in the fall to these nukes, we will, but these we are not treating. So these are daughters of our selected stock, but we're making divides. They have daughter queens in them, but we're not treating them. And these are survival stock, and we wrap them in groups of 3 to 5 nukes together, and we're wintering them like this without treatment. So their entrances here this is one with 3 in it, are facing south. They're wrapped with styrofoam insulation and some black plastic on the top. And they winter well for us like this. We're getting about 70% survival of these nukes. Not all of them survive. And especially without treatment, they won't survive very well. But why am I doing both? So I have these big parent colonies that I'm evaluating for many traits. And for right now, I'm treating them to keep them alive. And I have their daughters, which we're wintering and not treating, that I'm using as survival stock. My whole goal is I know that bees know what they're doing better than I know. And I would like to follow their lead. So the survival stock will tell me if they can survive. But as a scientist, I want to know why they're surviving. I want to know where they're leading me. So because we're measuring so many traits of these parent colonies, and I have all the pedigrees, and I have samples of all the bees, their virus, all of their characteristics, I can calculate the heritabilities of the daughter stock later, but I can also understand through the pedigree why some of these nucleus colonies are surviving and some not. And maybe I can trace it back to the genetics of the parent and then look at the characteristics of the parent colony to understand why they're surviving. So a lot of people do survival stock. We really don't have any idea or very little idea why they're surviving. And for me, I'm going to do survival stock, but I need to know, or I hope to know, why they're surviving. So I'll stop there. All of the publications that I referred to are on our website, which is here. It's blabumn.edu. And if you go to the Spivak Lab tab and click on publications, you can, if you have extreme insomnia, you can fall asleep reading our publications. So I think I'd like to stop sharing my screen if I can remember how.
I can talk at the same time.
Where's my shared window? Okay.
Up in the top.
Okay.
Okay. I'm even not able from where I'm sitting to see if your screen is shared. Thanks a lot. Wow, great. Really, really exciting and very, very important work that you're doing. We have lots of questions, or we have questions.
And you can see me now, right? It's
yeah,
okay?
we can. Yeah, you are on the screen on your own. Yeah. This is kind of longer questions, some of them. So there's one he's asking, you know, this Caucasian bees. Let's go to the propolis thing first, then we go back to the breeding afterwards.
Okay.
So there's a beekeeper from the southern part of Denmark. He says, okay, the Caucasian bees, they are very, very well known for being very, very good propolis producers. Does it mean that it's actually a better bee or a more healthy bee? Should we all switch to that one now, or what is your recommendation?
Right. I wish we had Caucasian bees here. We have Sue Kobe introduced some semen from Caucasian stock, and I have 2 Caucasian daughters from that. And they have a lot of propolis. They're not necessarily the best bee. I find good propolis collectors from the Carniolan bees that I have, and from some Italian derived bees that I have. So you can find good propolis collecting bees from in any stock or race of bees. I don't think you should switch bees completely. You can look for the propolis collectors. You know which ones they are. I think in the United States, for sure, we have over 100 years of selection against bees that collect propolis because we as beekeepers don't like it. But now I think through the research, we understand how important it is to the bees, and we missed the boat. I think we've made a big mistake, and we need to allow the bees to make this envelope for their health.
Yeah, I can give you a comment. It's the same case here in Denmark. And I think it was, I even do not think that the queen breeders did that on purpose, but they did select the ones that they have less propolis in, because then you could take out the frames with 2 fingers without any big mess, and you didn't get greasy fingers afterwards. So actually, I think also for the Danish breeds, we are very, very low in propolis production. And I'm in a group, we call it the Propolis Pollen Group, and we have over the last years tried to kind of find out how to collect propolis. My bees do not collect propolis. That's simply a case. But then luckily we find other ones, they produce pretty good. So that's very good. So this is putting me to another question. So that's Jesper. He's asking, could we kind of paint? I mean, you try to make the envelope, but why can't we harvest some propolis? Even maybe this tincture, or then paint the inside of the box?
Yes, you can. And that's how our research began. We actually collected propolis, and then we made a tincture, and we calculated the percent of we used a 15% solution, and we painted the inside of the boxes. It's just easier to let the bees do it, but that is one way you can do it. You would need to reapply it every year, I would think. But I think I know actually that after you apply it once, the propolis provides a sticky enough surface where the bees will add some propolis on top of that. So maybe it's a good way to start, but I prefer that the bees collect their own fresh local propolis, and I think that's probably the best for them. So that's my personal preference.
Oh, great, yeah. And now you get the question, and I know you will give the political correct answer. This really, again, the beekeeper asking. So what races should we prefer to use? Bee races? I know it's impossible to answer that question.
That's okay. It's a question that the beekeepers ask here all the time. And, I would say something like, well, you kind of use the American race, which is just a mixture of everything on the planet, is probably, it does. I think you use what you like, the bees that work for you, the bees that you prefer. I don't think there's one best race or stock of bees. They're the ones that you personally prefer and that survive well in your area and with your type of beekeeping operation. So, we have huge, enormous operations here in the United States, 40,000 bees, 20,000 colonies, huge. And they like particular kinds of bees that produce a lot of brood. And the more local, smaller beekeepers prefer Carniolan stock that are more conservative. So there's no one answer to that.
Yeah,
there's no best race, I don't think.
I totally agree with you. This is really a personal thing. How do you want to make your beekeeping? How how do you kind of treat your bees and so on. It's a very personal thing. So some bees fits to some beekeepers and others do not fit to them. So that's very clear. There was now we also you get the question all we got. I mean, what kind of bees do you have in the Minnesota hygienic hive? What kind of bees were that?
For the Minnesota hygienic line started out with Italian-derived bees, but then after a while, I would get, so they became a mixture. And in the United States, we mostly have a mixtures of Ligustica and Carnica, right? That's all we really have. And so they're all mixtures. And it became a mixture. Every year, for years, I would purchase 20 queens from different producers around the United States. And I would put them in a different apiary and I would test those colonies to see if I like them, if they survive the winter well. And the next year, if I like them, I would raise daughter queens from them and put them in the breeding program. If I didn't like them, I didn't. So I kept adding genetic diversity, different queen lines, and I was trying to maintain at least 10 queen sublines within the hygienic stock. Right now, in the new breeding program, I have 8 queen lines or 8 sublines that I'm really liking, and I hope to add more to make sure we have sufficient genetic diversity too.
Could you put some comments on the general American gene pool? How is it? I mean, we have heard in Europe that the concern in the US is actually that your gene pool for the whole US is very narrow, or how is that?
Actually, the gene pool is not so narrow, but the health is not good. So the genetic diversity is fairly good because there's a lot of queen producers and a lot of beekeepers that want different things. And so I think the genetic diversity is fairly good. Some researchers say that it's not so good, but all the research that I've seen has shown that it's really, the colonies are genetically diverse. But the health is really not very good. And it's because of the treatments that we apply, that we've always been applying, and the reluctance of the commercial queen producers to try survival stock or to really select bees for resistance. They can sell all the queens they produce. There's such a high demand for queen bees that they're just treating and selling and selling. And so diversity is good, but health is not so good.
So actually, when you talk about that you work together with some commercial queen breeders, this is only a very few ones, and is that the truth? And the other ones, they do it very conventional, as they always did. The queen breeding.
So most of the commercial queen producers that sell the majority of queens in the United States come from Northern California. And there's another group in Hawaii, and they're doing some good breeding on the island of Hawaii right now. It's excellent. The ones in Northern California, there's many of them, and I would say maybe a third of them are really selecting for hygienic behavior. However, they're all in the same area, and so queens are mating with the drones in the area, and so they're not able to saturate the area with good hygienic drones. So the queens are good hygienic stock. If I purchase a queen from them, I'm looking at their drones. I can use their drones, but the colony that I get will not necessarily be hygienic.
Okay. Nice. We have a German queen breeder in our group as well today. That's very nice that we share with other countries. It's Lutz. And I'm not sure if I do understand your question, Lutz, but he is writing directly like this. Is the propolis genetic also a recessive way? Does this sentence make sense for you?
I think he's saying, is it heritable? Is it
yeah.
Can you select? Yes, it's a genetic trait, like hygienic behavior, like swarming. It's a different.
Yeah, that's the way I understand it as well.
Yeah, so you can select colonies that have bring in a lot of propolis. You can raise queens from those, and it's selectable. Yes, I don't know if it's dominant, recessive. I don't know anything about how it's inherited.
Yeah. So maybe we should. I have the last question about the propolis, and actually, I have to say I was the one asking you this when you were making the talk in Sweden. What is your opinion about wooden boxes and the styrofoam boxes? And really, do you have an opinion about are we beekeepers actually cleaning us ourselves into problems? Because in Denmark we clean the boxes, with soap, the wooden ones are burned, and I mean we really want to have clean boxes. Is that a mistake?
I don't think it's a mistake. No, I think there's so much contamination with viruses and nosema and other issues that having the clean box is okay. But I think in addition, if you had a rough interior box, that would help clean, because the propolis is highly antimicrobial, and so that would help with the sanitation.
So hygienic behavior. This is so exciting, and I really regard the hygienic behavior, the freeze test, or the pin test, is really what I call a practical tool in practical queen breeding. We don't have all access to all those labs that you people have, the scientists and so here. There's one he's asking here, how long time would it take, where's the question? How long time will it take to go from non-hygienic colony to a hygienic colony when you requeen? Probably with the hygienic queen.
And well, about 2 months. So you need to have, the queen, the whole population needs to turn over. Okay. And the bees that are performing hygienic behavior are on average middle-aged bees from 15 to 21 days old. There's a lot of variation. So you need the adult bees in the colony to be from the hygienic queen before you'll see it. So about 2 months.
Okay, and actually now he is also asking a second question. It's really what happens if you're doing this by random mating?
Right, you can.
Yeah,
you can, but it takes a lot longer. Yeah. So I know in Argentina that I know a group down there that was only selecting from queens. Mm-hmm. But if you're in an isolated area and you can requeen colonies, and those will produce good hygienic drones, because if the queen is hygienic, her drones will be, right? And so if you're able to keep an area where there's a lot of good hygienic drones in the area, you can make good progress with hygienic behavior. In fact, this is what I'm trying to do now. I moved away from insemination because, for various reasons, one, my eyes are not, I'm older now and it's harder. But mostly, I became concerned about if I was introducing something with the inseminations. In other words, if I'm selecting for queens that are able to survive the insemination process, I mean, was insemination itself adding something that to these colonies or to the diversity of the colonies? I prefer that they naturally mate. So that's what I'm doing this time.
Oh, great. Just to go back to you talked about this new, I don't know, substance that this, I think it was a PhD student or something, is working on right now to spray on the on the brood. And I actually did hear that talk as well. And that's something that's under development. Somebody's asking what is the name of this product and where to get it.
Yeah, well, it's a product she's producing in her laboratory, and it's not for sale. I will tell you the compounds that this is from Olav Ruppel's old lab. He moved to Canada, but he was in North Carolina, and his student Kara Wagner, his PhD student, was the one who did the research. It's a blend of 3 or 4 compounds, I believe, but I'll tell you they're different compounds than the one that they found with Fanny Mondet and Yves LeConte and Alison Mercer. They're different compounds. And so although Kara is saying her compounds will help select for VSH, I think we need to have more research before we start selling these compounds, because I'm not sure hers are the best ones yet. Maybe the ones Fanny and Yves are finding in France, maybe those are better.
But for me, it's very encouraging that actually Stein is working on developing our testing kits. So
Yes.
we just have to sit down and wait maybe a year, 2, 3 years, who knows? Now there's a very exciting question. What can a backyard beekeeper actually do to produce hygienic colonies when we, and exactly he's the guy writing this, since we do not have access to artificial insemination or labs and so on. What is your, advices for the backyard beekeeper?
Well, don't forget the drones. That's what my advice. So if you test a colony for hygienic behavior, and that colony is hygienic, remember that you are doing a good service for your whole area because they're producing good drones. And so if you're in an area with a lot of other beekeepers and everybody is trying to use at least 1 or 2 hygienic colonies in the backyard, they're producing drones that will mate with neighboring queens, and it will help the population. So it's a community service to have good drones.
It's a community responsibility, I would say, actually. Yeah. So we have the queen breeder on one side. They really, we have science doing very nice projects and really working hard. And we have the queen breeders working very hard as well. But I think also, as you say, that we as backyard beekeepers also have a responsibility, instead of sitting, with crossed arms and waiting for you guys solving the problem. Actually, what you're saying there is the responsibility of the backyard beekeepers as well. There's one asking, and probably, are you a treatment-free beekeeper? No, you're not. What do you do with your colonies? When do you treat them?
My personal colony, I have 3 in my backyard, and then we have hundreds at the university. My personal colonies, if they have mites at the end of the season, I use in end of August, approximately after I remove the honey, I'll use formic acid. And if there's still mites, I'll use oxalic acid. And that's because where I live in the Twin Cities, there's thousands and thousands of beekeepers using highly susceptible stock. And my bees they're so close together, my bees might go rob out a colony that's dying and bring back mites, and then I lose my colony. So right now in our area, there's such highly susceptible stock I have to treat. But in the breeding program, in this area that I told you that's isolated, those nukes we're not treating. And we'll see how that goes. And I hope to then take stock from these and share with beekeepers in my area. I would prefer to not treat.
But you have to.
I have to.
Yeah. So now we have several questions from Christian in the southern part of Zealand, or Lolland-Falster. I have to be correct with the names, otherwise they get insulted. We talked about this genetic diversity in American bee strains, and you say there is actually a good diversity. But since we have, is it expected with the bees in Europe, Asia, and Africa that it should be expected that they already have the hygienic behavior? Naturally, so that you don't have to breed for it. You just have to go and get those bees and maybe cross them into your own lines.
Yes. I think when I first started looking for or doing the freeze-killed brood assay in the 90s, I found about 10% of the colonies that I inspected showed good hygienic behavior. So if I tested 100 colonies, I would always find 10 from any stock of bees, from any producer across the United States. Now it's closer to 20% because beekeepers are starting to raise more queens that are from hygienic stock. It's easier to find these colonies. So about 15 to 20% of all the bees, I think, in the United States now are hygienic, which is good. It's not enough. It's a slow percentage, low, but it's better. And I think if you go out and look, you'll find them and you just propagate those.
Mm-hmm. We have a nice comment here from Susanne, and I think she's from Norway. And up in Norway, they have this, they have those colonies that have not been treated for many, many, many years.
Yes.
And she's actually talking about that. Is it really like this, that if you have raw tolerant hives, that if you kind of switch those hives to your local bees or your local apiary, that the homecoming foragers bees will kind of be taught, or do the bees teach each other to become tolerant?
I wish they did. I wish they did, but they don't.
Okay.
It's genetics that's mostly controlling that tolerance. And so I'm going to talk about this for a minute. So one of my fears or concerns when I'm doing this new breeding program and I'm in an isolated area, I know that for miles there's no other beekeeper. And so I can do these naturally matings, and I know, or I hope, but I know that within 4 or 5 years, I'm going to have stock that I don't need to treat. The parent colonies, none of them. I think we can go without treatment because they will be good survival stock. However, when I take those queens here, where I live right now, into the city or an area where there's a lot of beekeepers, I'm afraid those queens won't still retain their tolerance because of robbing and drifting. Because of the high mite pressure in this area, those colonies may still collapse from mites and viruses. So this is what my concern is that we can find these good resistant stocks or tolerant stocks, whatever you want to call them, on islands and in Norway and sometimes in France and in the upstate New York and hopefully in my area. But if you take them away from those areas, do they retain their resistance? And that's an issue. We really need to be looking for this on a much larger scale than locally.
Yeah, I think that's really the point, because of course we have tried to buy queens from queen breeders that, you know, claim that they're raw tolerant and on some level, and then we take them into the Danish bees, and there's really no difference. Could that really be because the types of varroa mites are different types of varroa mites, subtypes of?
I don't know about types of varroa mites, but I think it's a lot of it's a virus issue.
Yeah,
that's why we're also looking at viruses, because we need to also be selecting for bees that have and retain low virus loads, but it's extremely expensive and extremely difficult data to obtain.
And maybe give a comment again on Denmark and Norway. I mean, in Norway they treat far less than we do here in Denmark, and on some surveys they really found out that they have less virus and virus load in Norway than we actually have in Denmark. We have far more viruses in Denmark, and therefore we really have to treat, I would say, even far more. Yeah.
Right. Well, this is what's going to happen. If you're able to select for varroa tolerance, your virus loads are also going to go down. Fewer mites, less virus load, and maybe different virus variants. So this is work Declan Schroeder does, that after a time, the variants change. I mean, we're learning this with COVID now, right? With all the different variants, some were easily transmittable, some not so. This is the same with DWV in honeybee deformed wing virus, for sure, that we know there's many different variants and some more virulent than others, and that virulence changes in different bee populations. But over time, if we can get the mites level, if we're not using so many treatments, the virus levels will go back down too. A virus does not want to kill the whole colony, because then it doesn't have anywhere to go. Same with
I mean, a parasite does not want to kill its host. That's killing itself.
Right.
I have to jump fast back to the propolis thing again. This is Camilla asking, would the bees naturally cover rough surfaces with more propolis than smooth surfaces?
Yes, that's the thing. Our boxes that we purchase are very smooth. They're planed smooth. If they were unfinished lumber, the bees would attach propolis to the inside of the boxes, but not very much. If it's really rough, they really put a lot of propolis in there. Not all colonies, so some of your colonies, Fleming, may not.
Yeah, but I also have styrofoam boxes, so, and I clean every year, so maybe I have to stop. This is Christian here asking how many survivor families or colonies do you winter every year?
Right. This winter we had 75 survival nucleus colonies. 76, I think, we wintered. And we have right now about 80 of the big parent colonies. That's how many we put into winter. I haven't even checked to see how many are alive yet. Next week it's supposed to be warm enough, maybe we can check. So I don't know how many survived.
Okay,
About
it's
100, about 150 total.
Yeah, it's always very exciting.
Mm.
Per, he is asking if you have a hygienic colony, do you dare to guess what fraction of the bees in that colony are the ones taking care of the hygiene
Yeah, I do
behavior?
I do dare guess. And it's a fairly small percentage. First of all, there's only a certain group of age of bees that do the behavior. So the foragers and the very young nurse bees are not the ones doing the hygienic behavior. It's mostly that middle-aged bees. So if they compose 25% of, or 35, I don't know what percent they are, and among those, it's only a relatively small proportion, but I can't tell you exactly how many. It's a small proportion of the bees that are actually doing all the full uncapping and removing. There's more bees that do the uncapping, and then more bees that come and recap. And I believe the recapping is because they can't detect that there's a problem. So I think some bees are good at detecting and the others recapping. They are finding that this recapping behavior is associated with lower mite loads. But I think it's because some bees, there's enough hygienic bees in there that are uncapping in the first place that they're able to find where those mites are reproducing and disrupt that reproduction somehow, I think.
Yeah, this is one of the beekeepers asking if you do not treat them, then there will be a natural selection for more tolerant or resistant bees. But what do you do? I mean, do you just accept that those ones, they will die out, or do you have a threshold and then you say, okay, now we treat and it's out of the breeding program? Or do you go all the way? Natural beekeeping.
That's why in this new breeding program I'm doing both.
Okay.
For the nukes, they're going all the way. They're on their own. If you survive, you survive. If you don't survive, you don't. But I'm holding on to their mother colony. I'm holding on to their parent because I want to know, and I want to keep those colonies alive to understand their characteristics. So for me, letting them all, the survival stock, just letting that many colonies die is hard for me. But it also, I know, is an extreme genetic bottleneck. It reduces the genetic variability, which I think is not a healthy strategy, personally. So, yeah. So I'm doing both. Did I just see Tom Seeley flash on the screen?
Yeah, you did. And it made me very happy because the next speaker he's in the system, and we will not have a disaster of people not turning up.
No, that's good. Tom Seeley and I end up at the same conferences a lot recently.
Yeah, you're the hot topics at the moment.
Oh, are we? Isn't that fun, Tom?
So there's one asking now, and maybe this is again hypothetical. How long time will it take before we have bee lines that can clean out close to 100% of all diseases? Is that a possibility to kind of make a statement on that?
No, it's impossible to make that statement because I can't control human behavior. But actually, I would even say, I'll use Tom Seeley as an example. If you go into the Arnott Forest, in that group of colonies that are able to live with Varroa, if we were able to get into those trees and test them for hygienic behavior, not all of them would be hygienic. I think that's one of the traits that bees are using to survive with varroa mites or tolerate varroa mites. I think there's a number of traits, including life history traits. And this is kind of a segue into probably what Tom's going to talk about. It's the whole characteristics of the whole colony. How big are they? How much brood do they have? How often do they swarm? What are they doing to keep the mite levels lower just through their natural life history characteristics? So just having one trait, hygienic behavior, for me, when I was selecting for the hygienic line and just selecting for that one trait, it was not enough for the bees to be resistant. I still needed to treat.
Could you give some comments? It's really one asking here, what is the, I mean, we have heard about this very, very high winter losses that you have, or even summer and winter losses that you have in the US.
Yeah.
But what would the normal losses be if we excluded the almond colonies that kind of go to our almond pollination? We would expect that for us, we would expect that would be I mean, what would be the normal winter losses without this very high level of migration to California?
Actually, the beekeepers that take their bees to almonds have higher survivorship in their colonies than the backyard beekeepers. So they're treating their colonies a lot. They're they do have some operations experience extreme losses some years, not every year. But in general, the commercial migratory beekeepers have higher survivorship of their colonies compared to many of the backyard beekeepers. And it's because they're treating so frequently. But I think our average colony loss in the US is 30–40%.
That's a lot.
It is a lot.
That's a lot. Christian, he's asking you, isn't there two kind of hygienic bees? I mean, some bees are uncapping and other bees are kind of removing the mites or removing the larvae.
Well, I think it's part of the same motor. It's a behavioral trait, and it's a motor pattern that begins with detection with their antennae, and that then some bees that detect, they're like curious, if I could attribute human characters, and they, what's wrong? And they open up poke a hole in the cell capping. And if the stimulus coming from that pupa is strong enough for them to detect, and yes, there's something wrong, they continue to expand that hole and then continue the process of removing. We know from previous research that the bees that do the uncapping have higher olfactory sensitivity than the bees that do the removal process. So the most sensitive bees, those with highest olfactory sensitivity, are doing the uncapping. And then once they expose this brood that's abnormal for whatever reason, other bees are able to perceive or detect that there's something wrong, and they'll continue with the removal process. So it's really part of the same behavioral repertoire, the whole sequence of events.
A nice question from our colleagues in Sweden is actually asking, it's about chalk brood, and he's kind of hygienic behavior in regard to chalk brood. How do we measure this when we don't see the chalk brood anymore in the frames, or if we see the chalk brood laying on the bottom board?
Well, if you see the chalk brood on the bottom board, it means the bees were detecting and removing it, but too late. They were removing it when it's infectious, and that risks transmitting the disease, right? Of course, they can be removing some larvae with very, very early stages of chalk brood that you barely can see without with your eye. You'd have to see in the microscope. And if they remove those and they stay on the bottom board, they will develop into mummies. So you really don't know when it was removed, but generally bees are removing, if you find a lot of mummies on the bottom board, they were removing it after they reached that infectious stage. So you really want colonies where you're not seeing chalk brood mummies on the bottom board.
So could you give some comments on I would say local breeds, bees that are fitted into the local climate, local area, the local biotopes? I mean, what we're doing right now is actually, if I look on the US, I mean, you send queens from California to to Wisconsin or something like that. This is bigger than Europe. So what do you mean? What do you think about having local bees, focusing on local bees? Is that something we should do, or is it just?
Well, I would never say anybody should or should not do anything, but I would think that would be a wonderful idea. And there's kind of a movement, a grassroots movement of beekeepers within the United States to create more local breeding programs, to not rely on queens that come from California or from other states and not rely on package bees, the sale of package bees, to winter these nukes like I was showing, and then sell the nukes or sell queens from those nukes on a local scale so that we could have locally adapted stocks. I think this is a wonderful idea. I would really like to expand this for northern climate beekeeping in our area, and to have beekeepers that would be doing this collectively. Could I control the behavior of all the beekeepers in my area? No, and that's the problem.
Yeah, but it's also sometimes also the fascinating thing about beekeepers, that we are the viability is really, our variation is really big. There's one asking about it. He got confused about your varroa treatment. I mean, you give formic acid late in the season after the honey harvest or something like that.
Yes.
And then you give an oxalic acid trickling maybe around, I don't know, late in the season, Christmas.
For us, it would have to be October.
Okay.
By Christmas, we can't open the colonies.
Okay.
But yeah.
Marla, we're getting to the end. This was very fascinating, but now you get a very philosophical question.
Oh boy.
Can the humans learn something from the social bees when it comes to fight COVID or Corona? Is there something we can learn from that?
I think we can have empathy. I hope we learn from the bees, but I think we can have some empathy for what our bees are going through right now with these viruses. And in fact, okay, I'll give you an example. I had my second COVID vaccine last Monday. Oh. And the next day I had a fever and I had chills and aches and pains. And as I was on my couch in my living room, I kept thinking, oh my goodness, sometimes in our experiments at the university, we inject bees with an immune elicitor. It's a bacteria that's heat killed that doesn't produce disease, but it creates an immune response in the bees so that we can measure how their immune system's working. And I was thinking, wow, what I feel like is what my bees must feel like after we've given them these injections, because my body is producing this huge immune response to the COVID vaccine. And I started wondering if this is really ethical, what I'm doing to give the bees. But that's just a side story. I'm glad to be vaccinated.
Yeah,
it's really important for everybody to understand. I think humans are starting to understand disease transmission, virus transmission in dense populations, and what our social insects colonies confront every day of their lives within a colony. How can they prevent this disease transmission or virus transmission? And when they have this virus, what it means for their health. So, and we can't put masks on the bees, and they can't social distance within the colony, but they can certainly social distance among colonies. What does this mean for colonies kept in really dense apiaries, which I think Tom Seeley will address? Or commercial operations, or cities like where I live, where there's super dense populations of bees and beekeepers. This does not bode well for colony health. So thank you for the question.
Great, thanks a lot, Marla. Now you can go get your breakfast. You can get a cup of coffee, and I hope to see you next week. Thanks a lot. Lots of good reaction here in the chat, and great talk. I'm really happy that we managed to do it.
All right.
See you next time.
All right. Thank you so much.
Bye-bye.
Thank you, everybody.